Dear learners, welcome you all in the course of Immunology BZYET 141. Basically, it is a elective courses for BSc General. And dear learners, my name is Dr. Siaram. I am an associate professor, Discipline of Life Science, School of Sciences, Ignou. Dear learner, today I want to discuss very important topic of this course, that is known as the antigen antibody interactions. It is a very important chapter of your courses. Dear learners, what is the expected learning outcome of today's session? In this session, we will discuss the various things. Dear learner, after completion of this session, you will be able to define the antigen antibody interactions. You will be able to describe the mechanism of antigen antibody interactions. And after completion of this session, you will be able to explain the concept of immunological technique which can use for diagnosis of the antigen concentration or antibody concentration. After finishing of this session, you will be able to discuss how immunological technique used for detection of the pathogens. And finally, you will be able to explain the advantages and disadvantages of the immunological technique. So it is a expected learning outcome of today's session. So my session is divided into two parts, part first and part second. In part first, we will discuss the various things like the principle of the antigen antibody interaction, like precipitation, immunoprecipitation, immunodiffusions, and neutralizations, and complement fixations and opsonization. So these are the various mechanism we will discuss in detail. While in part second we will discuss the various tools and techniques we can utilize for diagnosis of the antigen antibody concentration, like Western blotting, flow cytometry, like ELISA, like radial immunodiffusions, various technique we will discuss in the part second. So I want to discuss one by one in part first. In this slide, I want to discuss what is the antigen antibody interaction. The antigen antibody interaction provided an example of the epitope paratope interaction. Epitope which are available into the antigen, and paratope which are available in the antibody. They will interact to each other. Then it will neutralize the pathogen, neutralize the antigens. It is similar to enzyme and substrate reaction. The paratope and epitope interaction, it is just like the enzyme and substrate reactions. The antigen antibody interaction are very weak. Their bonding between the antigen antibody, it would be very weak. Like hydrogen bond, like van der Waals forces, various weak interactions are there, and these bonds are species specific. Basically, when the antigen antibody it binds, they have the specific species like CH3 group, OH group, and NH3 group. Various species are there. Ultimately, the antigen antibody, they will bind together, or ultimately they are able to neutralize the reaction. And these interaction including hydrogen bond, ionic bonding, and hydrophobic bondings, and interaction between van der Waals rings. You can see in my figure, here is the antigen antibody interaction. It is a epitope which are available on the antigen, the various 3D structure which are available onto the epitopes. It is a paratopes. The paratopes are there which are available on the antibodies. It is the molecular interaction, the various bonds which are available in the paratopes and epitope. They will bind together, like hydrogen bonds are there. You can see the bond between H and OH. It is the hydrogen bonds. Another is the ionic bonds will be there in the antigen and antibody interaction, like NH3 and O bonds are there. So they are the ionic bonds are there. So ultimately they will bind together or they will neutralize the antigens. Hydrophobic interactions are there. You can see the structure of the hydrophobic interaction between the antigen and antibody. And van der Waals interactions are there. So these type of interaction which are involved within the antigen antibody. So you can say it is a molecular interactions which are very important. They are able to neutralize, they are able to eliminate the pathogen, eliminate the antigen which are available inside to the body or outside of the body. So here is the different interaction between the antigen and antibody. In case of antibody, they have the paratopes, and while the in case of antigen they have the epitopes. They have the several species are there, or ultimately they will bind together. So you can say it is a antigen antibody interaction, or molecular interactions are there. What is the general feature of antigen antibody interaction, which are very important? Individual species behave differently, like NH3, CHO, OH group, and number of the other groups which are available on the paratope and epitopes are there. They behave differently, and each antigen and antibody molecule participate in these reactions, and no denaturation will occur while the reaction taking place. If antigen and antibody will bind together, no denaturation will occur. And antigen antibody are basically basically the superficial type of the interactions are there, and the combination can be reverse, and the binding of the antigen antibody can be reverse type of the reactions are there. Interaction is directly proportional to number of the valencies. Number of the valencies in the antibodies, like one antibodies, they have the two valency are there in case of the IgG and IgA. But in case of the IgM, they have the 5 or 10 valencies are there. So it is the interaction is directly proportional to the number of the valencies which are available on the antibodies or even antigen are there. After that, I want to discuss the mechanism of antigen antibody interaction. There are the several mechanisms are there. We will discuss in detail. The first mechanism, immunoprecipitations. Another is the agglutination, and third one is the neutralization reaction, and fourth one is the complement fixation, and fifth one is the opsonizations. So these are the various mechanisms which are involved in the antigen antibody interaction. I want to discuss one by one, like immunoprecipitations. Immunoprecipitation, when antibody react with the soluble antigens, a lattice is formed which later converted into visible precipitate, and this lattice formation is depend on the valency of the antigen and antibody. The valency is not only present into the antibodies, but it is available in available on the antigens. So it totally depends on the formation of the bond between the antigen and antibody. The antigen antibody must be bivalent or multivalent are there, and the precipitation curve represent a bell shape. You can see this curve. Here is the antibody in precipitations. Here is the increase of the precipitations are there. Here is the antigen added. If you antigen added, the precipitation will start. Here is the zone of excess of the antibodies. The very less reactions are there. Here is the antigen antibody are equal. You can say that is the precipitation in the zone of equivalence. Here is the antigen antibody which are equal. They will bind together. Their concentration it would be same. Here is the zone of the excess of the antigens are there. So you will get the bell shaped curve of the immunoprecipitation. So these are the very important curve it need to understand. Another thing I want to discuss the immunoprecipitation in the sol. Basically, colloidal solutions are there of the precipitations. This method is used for to the quantitative estimation of the precipitate. So we can utilize for the quantitative detection of the sol. In the different test tube, antibody are present into the equal quantities. In each test tube, antigen will be added in successive increase of the amount. After precipitation, centrifugation is done and found to the standard precipitation curve. Then we can decide it the actual concentration of your antigen or actual concentration of your antibody or there. So it is a immunoprecipitation of the sol. Another is the immunoprecipitation in gel. We can utilize the gel to measure the immunoprecipitation between the antigen and antibody. In this method, antibody is embedded in the agar medium. So we can take the agar medium, we can make a gel, we can use the antibodies, we can edit the antibodies in the gels. When the antigen is diffuse, a visible line appear, the amount of antigen antibody equal. It is formed by it is performed by two method, Mancini method and Auster-Loney method. So these are the two methods we can utilize it for detection of the concentration of the antigens. And third one is the co-immunoprecipitations. It is a modification of the immunoprecipitation technique. So we can modify the immunoprecipitation techniques are there, that is known as the co-immunoprecipitation. In this case, the antigen we can utilize it. First of all, it need to lyse the cells, and various procedure we can utilize it. Or you can say the various step we can utilize it. The first step is the preparation of the cell lysate. Here is the cell lysate, we can prepare it. You can see it, here is the cells. We can make a lysate. Binding of the bed and prey of the GFP trap. So GFP trap we can utilize it. Here is the antibodies are there. It is a GFP trap. Washing of unbound molecule, prey should remain bound to the bed. An illusion of the bound bait or prey from this bait. Analysis of the weight or prey onto the SDS page, Western blot, or by mass spectrometry. So these are the procedure we can utilize it for the co-immunoprecipitation. Ultimately, we can purify of your unknown concentration of your proteins. Here is the various steps are there: cells, cell lysis, cell extract, and antigen antibody complex, antigen antibody. Here is the bait complexes are there. Ultimately, you will get the pellet. We can analyze it. Second one is the agglutination reactions. So what is the mechanism behind the agglutination reaction? In this method, the granular antigens are used instead of the soluble antigens. In earlier techniques, here is the we can utilize the soluble antigens. Here is the granular antigens, we can utilize it in case of the agglutination reaction. The antibodies which shows these reaction are called the agglutinin. In excess of the antibody, precipitation reaction blocked, and similarly the agglutination reaction inhibited, in such type of the effect is called the prozone effect. Agglutination is of the two type. One is the direct agglutination, another is the indirect agglutination. So you can classify it into two ways. One is the direct agglutination and second is the indirect agglutination. So we can understand the direct agglutination. It is used to detect of the large size antigen like RBCs, bacteria, fungi in case of the direct agglutination. It is directly bound to the particular pathogen, RBC, fungi are there. So they can bind it or we can measure it their concentration. And it is performed into the test tube or a small cavities. In each cavity, the amount of antigen is the constant, and but serum containing antibodies is gradually diluted 1 ratio 20, 1 to 40, 1 to 80, 1 to 160, and we can vary the concentration of the antibodies gradually. An example of ABO blood group using for the typing. Another is the RH antigens. So we can measure it with the help of the agglutination test, like direct agglutination test. We can measure the different type of the blood groups which are available in the humans, and we can also measure the RH antigen with the help of the direct agglutinations. Another is the indirect agglutination, or you can say the passive agglutinations are there. It is a very sensitive method for antibody detections, and RBC, bacterial cell or inert particles such as the latex can be used as a carrier for to the antigens. In case of the direct agglutination, we cannot utilize the carrier, but in case of the indirect agglutination or passive agglutination, we can utilize the carrier molecules like latex. We can utilize it for the agglutination reaction. This make the reaction more visible. We can visualize under the light microscope. Even we can visualize through the naked eyes. If any agglutination reaction will take place, we can see through the naked eyes. The latex agglutination, RBC, passive agglutinations are there. If the antigen is attached to the particular carriers, called the reverse or passive agglutinations. Rheumatoid factors detection, C-reactive proteins detection, CRP detections are there. We can utilize for detection of the various tests are there, like CRP, C-reactive protein we can utilize for detection with the help of the indirect agglutination or passive agglutination. The type of agglutination reaction, one we can utilize in the glass slide. We can utilize the test tube, we can utilize the microtiter plates in passive agglutination test or indirect agglutination test. So there are the three type of procedure available, medium or available, like glass slide and tubes and microtiter plate we can utilize for detection of the different type of the protein, like C-reactive proteins are there. And third one is the neutralization reaction. It is a third very important mechanism of the antigen antibody reactions are there, or antigen antibody interactions are there. The neutralization reactions. It is a process by which the specific antibody neutralize the bacterial or any viral toxins are there. So we can neutralize with the help of the antibodies like pathogens like bacteria or viruses are they are the exotoxin we can neutralize with the help of the antibody. It is used for the diagnosis and treatment of the various diseases, and such molecules are called the antitoxin, which are particularly antibodies. The passive immunity can be provided by the infecting, such as the antitoxin in the human diphtheria and botulism. We can utilize the antibodies, or we can give the passive immunity, or ultimately we can save from the particular infections. Here is the neutralization of the viral particle by the serum antibodies. So you can see it. Here is the virus infected cells. They have the epitopes are there. The various epitopes which are present on the viral particle, you can say it is a pathogen. Here is the neutralizing antibodies are there. Neutralizing antibodies. They will bind on a particular epitopes which are present on the surface of the viruses, or ultimately the reaction will take place, or it will neutralize the various epitope. They will neutralize with the help of the neutralizing antibodies. The antibody protect against the pathogen in 4 ways. First one is the neutralization, second is the opsonization, and third one is the complement fixation, and fourth one is the antibody dependent cell mediated cytotoxicity. So there are the 4 mechanisms are there. Ultimately we can kill the pathogen, we can neutralize the pathogens, or we can get rid of from the particular pathogens are there. There are the 4 mechanisms are there. All the 4 mechanisms it require the antibodies. Without antibodies this mechanism this will not work, this process will not work, like neutralization, opsonization, complement fixations, and ADCC, it will not work. So there are the 4 mechanisms which are involved in the neutralization of the antigens or viral particle by the serum antibodies. And fourth one is the complement fixation, or you can say the complement systems are there. It is another very important mechanism of the antigen antibody interaction. It is a class of the plasma proteins which are available in the serums. They can impact on the immune systems. This system has the 11 variables factors ranging from C1 to C9. There are the several factors, several proteins which are available in the serum, that is known as the complement proteins, or complement factors are there, complement proteins are there. Factors such as the BDHINPP are the properdins factors are there. These factors work as a cascade flow, and they are found into the inactive form of the blood plasma and body fluid. And the primary proteins in this system is C3, which is triggered by the 2 route. One is the classical pathway, another is the alternate pathway. The classical pathway, the immune system is converted into C3 activations. In alternate pathway, polysaccharide on the surface of the microbe that activates C3. So you can see it is a cascade of type of the reactions are there. But here is very important things are there. Here is antibody which are involved. The antigens, they have the it is the pathogens, invading pathogens are, they have the antigens, or you can say they have the epitopes are there. So antibody which are bound with the particular epitopes. So here is important role of the antigen antibody interactions are there. Without antigen antibody interaction, the complement system will not activate, or ultimately they will form the MAC. Here is the MAC are there. membrane attacking systems are there. So they can attack it, ultimately pore will create or it will die. So these are the series of the reactions are there. Several factors which are involved in the complement fixes complement systems. Uh, there are the several steps are there. This system perform it action it three ways. One is the activation of the inflammation, opsonization, and cytolysis. So there are the three mechanisms which are involved behind this, or ultimately they are able to kill the pathogens, or they are able to kill the antigens. Another thing I want to discuss here is the complement fixations are there. It is a very important mechanisms are there. The complement fixation. The complement fixation is a traditional laboratory diagnostic test that is still used during the acute illness to determine the viral antibody in the patient's sera or cerebrospinal fluid sample. The test primarily assess the IgG antibody. You can see it here is the antigens are there. It is a serum without antibodies. Here is the serum with the antibodies. You can see it here is the hemolysis will take place in your reaction. Here is if complement is fixed with the particular antibody, hemolysis will not take place. So you can see here is the very important test are there. We can check the complement fixations, which are very important tests are there. They can check it the excess of the IgG antibodies in the serums. And it is the opsonization. Opsonization, it is another very important mechanism are there. It is a process in which the microbes that contact by the antibodies and complement protein. These are the identified by the phagocytosis and they digested. You can see here is the antigens, and it is the antibodies are there, they will bind together. Here is the complement B proteins are there. Basically opsonization, it help in the isonization your antibodies. Here is the antibody IgG1, IgG3 and IgM antibodies are main opsonizing protein. In CH3 region of these antibodies where active site are present for binding of the Fc receptor present on the surface of the macrophages. Here is the macrophages are there. They are able to bind with the antigen antibody reactions. Then opsonization will take place. The IgG and IgM activates the complement system by classical pathway or by alternate pathway. Due to this C3B convertase enzyme integrates C3B opsonin fragment present on the surface of the microbes. And this opsonization is facilitated by classical pathway as well as alternate pathway. Dear learner, I want to summarize part first. You have learned the various things. I want to discuss one by one. Antibody react with the antigen at a specific binding site like paratope versus epitope, forming reversible non-covalent bond at optimum condition. When the antigen antibody concentration are equals, they are able to form the complex, or you can say the precipitate. It can be purified from the mixture of the heterogeneous soluble molecules. So we can utilize as a co-immunoprecipitate. The immunological complex formed during the antigen antibody, it can be measured by the immunoprecipitation, agglutination reaction, neutralization reaction, complement fixation and opsonization. Dear learner, we will take a short break. We will come within 2 to 3 minutes, and don't go away.
Do you wish to be part of a television studio like this, or any other media system, or already part of any media system, want to enhance a new set of skills? Welcome to School of Journalism and New Media Studies. Here in the School of Journalism and New Media Studies, we are offering program for all set of people to learn and enhance their media skills. The vision of School of Journalism and New Media Studies is to generate a new set of professionals in the area of media and communication studies with understanding about role of media in the national development and global understanding. We offer innovative programs in the field of journalism and mass communication that sets the benchmark of high quality learning and teaching process at the national level. We offer these programs across the country with the help of our regional network as well as study center. Same manner, many of our programs are offered through online mode with a sophisticated learning management system. SOGNMS offers programs from PhD in journalism and mass communication till certificate level. We have masters in journalism and mass communication. The eligibility is bachelor's degree, and we are introducing this program in 3 languages: Hindi, Tamil and English. Students from national and international students can join either through online mode as well as in the distance mode. We have 4 postgraduate diploma programs, specialized areas in the field of development communication, digital media, electronic media and journalism and mass communication program. And all these 4 programs are available in distance mode. 2 programs are available in online mode, development communication and digital media. And we have specialized certificate program in community radio. Ignou employs various media and communication tools for better teaching and learning process. School of Journalism and New Media Studies utilizes this media system for delivering all our programs. Come and join with us for exploring a new career and enhance your media skills.
Dear learners, welcome you all in my part 2nd session. In this session, I want to discuss the basic tools and techniques, and these techniques we can utilize for detection of the antigen antibody interaction or detection of the concentration of the antigen or antibodies. So there are the various tools we can utilize it for detection of the antigens or antibody. The first one is the radial amino diffusion, or you can say the Mancini immunodiffusion, Ochter-Loney diffusion, double diffusion, and third one is the hemaggglutinations in Western blotting, ELISA, and immunofluorescence, immunoelectrophoresis, and flow cytometry. So there are the several tools we can utilize for detection of the antigen or antibodies. So I want to discuss one by one. First one is the radial immunodiffusion, or we can say the Mancini methods. The antibodies are the mixtures in the agar gels and poured onto the glass slide or petri plate, and bell are cut on the surface of the gel, and antigen is loaded. An antigen diffuse radially and form to the concentric ring of the precipitations, And concentration of antigen can be estimated by the diameter of the hollows, and we can utilize it for the detection of the various pathogen like influenza viruses, and we can estimate Ig class in the sera and different immunoglobulin classes in the sera. So we can utilize the radial immunodiffusion by Menceni methods. So you can see it in the figure. Here is the plates are there, Petri plates. We can utilize the antibodies in the gels, and we can put the antigen in the centers, and antigen antibody they will diffuse, or ultimately they are able to form the rings. In another figure you can see it, here is the rings are there. The holes are there. In this hole we can put the antigens, and they are able to form the rings around the holes. And the presence of the precipitin ring around to the antigen belt indicate the specific antigen antibody interactions are there. So here is the antigen antibody interaction is very much. Here is the very less antigen antibody interactions are there. The absence of the precipitin ring suggests an absence of the reactions, and greater the amount of antigen in the bell, the further the ring will be formed in the bells. Another is the Auster-Loney double diffusion method. It is based upon to the simultaneous application of antigen and antibody in separate but adjacent bell on an agar plates. So you can see it, here is the 90 plates are there. You can put the agars, you can create the bells. In this agar we can utilize the antibodies. As the material diffuse toward one another, and precipitation line form resulting from the antigen antibody interaction. So you can put your antigens, the antibodies which are available in the bells, and the precipitation ring forms. If the multiple bell of the antigen are positioned around to the antibody bell in the same plate, and several pattern of reactivity may be observed. The several pattern we can observe it in the Auster-Loney double diffusion techniques. You can see it in this result. Here is the precipitation zone form. The precipitation zone form after the radial immunodiffusion. Here is the precipitation rings are there. Here is you can see it, here is the precipitation lines are there. The serum with the A and C showing the possible for the pathogens. Here is the B bell, the no precipitation rings are there, the control with the pathogens are there. Here is the precipitin lines are, here is gel. So you can see it, the different pattern you will get. Precipitation line formed after Auster-Loney double diffusion method. You can see it, here is the identity. You can put the antigens 1, antigens 2, here is the antibodies are there. So you will get arc. Here is the, so you can say the identity are there. A related antigen fuse precipitate line, and no related antigens cross line. So you will get the cross lines, no identity are there. They are not able to bind with the antigen 1 or antigen 2. No relation of the particular antigen antibody are there. You will get the cross line. Here is the partial identity are there. Spur formation are there. So you can see it, here is the spur formation. Spore formation will take place. So here is the partial identity or against the particular antigens. Here is the antibodies are there. So you will get this type of pattern, or ultimately we can see it, the antigen is they have the identity antibody they have identity against the particular antigens. The antibody they have no identity are there, you will get the cross line, and partial identity you will get the different type of the patterns are there. Another test is the hemoagglutination test. The red blood cells can also absorb many antigens, and when the mix with the specific antibodies will form the clump and passive hemoagglutination. Red blood cell are passive carriers, and this can be inhibited by the specific antiviral antibodies. So you can see it in this slide. Hemoagglutination assay using antigen antibody reaction. Here is the antigen antibody RBCs are there. You can see it, hemoagglutination will take place. Here is the control buffer are there. In this sample the hemoagglutination will not take place. So hemoagglutination, it is a very very important. We can mix the antigen antibody, or ultimately clump will form. And fourth one is the Western blotting or immunoblottings. The Western blotting is an analytical technique widely used to detect, identity or quantify a specific protein in a complex mixture. And the Western blotting is also called the immunoblottings. And since the antibodies are used as a probe to detect the specific antigen, the antigen, it may be proteins are there which are available, particular proteins which are available in the cell are not, we can detect through the Western blotting or immunoblot technique are there. We can detect, we can identify, we can quantify a specific protein in a complex mixtures. This technique mainly provided the following major step. There are the 5 steps which are involved in the Western blottings. First one is the protein separation through the SDS page. SDS page are there. It is a techniques we can utilize it, or ultimately we can separate your proteins or antigens are there. The protein transfer from the gel to the membranes. So here is the protein we can transfer into the particular membranes are there. The prevention of the non-specific binding through the washing. We can wash it in the particular buffers, or ultimately the unbound proteins we can remove it. Then we can utilize the antibodies, antibody probing, that is known as the antibody probing. The antibodies, they can bind to the specific antigen, and ultimately we can detect the particular protein which are available in the membranes, or which are available in the peroxisome, or which are available into the different part of the bacteria are there. So it is a very important technique. We can utilize it for detection of the particular antigen or particular proteins. And another technique that is known as the ELISA, enzyme linked immunosorbent assay. ELISA, it is a well documented techniques are there. We can utilize for detection of the antigens or antibodies, and use as a immune reaction like RIA, radial immunodiffusions. It differ from the RIA and detect based on to the enzyme catalyzed reaction or fluorescent probes. So in this case we can utilize the enzyme catalyzed reaction, or fluorescent probe we can utilize it. An example of enzyme labeled probes are there. The enzymes, several enzymes we can utilize in the ELISA. One is the horse radish peroxidase. Another is the alkaline phosphatase and glucose oxidase. These are the 3 enzymes we can utilize in the ELISA. What is the advantage of the ELISA? It is very sensitive, nanogram level are lower and reproducible, and minimal reagent will take for the ELISA, and qualitative and quantitative a reaction will take place, like example of the qualitative, we can detect the HIV. In quantitative assay, we can utilize the drug monitoring. The greater scope well can be coated with to the antigen or antibodies. So in this case, well we can utilize it which are available in the tube or which are available into the 96 well plate. We can coat it either antigen or antibody. The suitable for automations. So your ELISA is the very suitable for the automation, high speed, no radiation hazards are there like RIA. The type of ELISA, there are the 4 type of ELISA are there. I want to discuss one by one. First one is the direct ELISA. You can see it here is the primary antibody conjugate. Here is the antigens, they will bind it or ultimately we can detect it. So in this case the antigen is immobilized directly on the plate. The antigens we can immobilize on the plate are microtiter plates, and the conjugated detection antibody bind to the targets. The added of the substrate produce a signal proportional to the amount of the antigen in the samples. So we can measure it through the ELISA plate reader. How much antigen are present, we can quantify it. And another is the indirect ELISA. You can see it here is the indirect ELISA are there. So in this case you can see it in this figure. The antigens are there. Here is primary antibodies which are not conjugated. The conjugated with the particular enzymes or fluorescent probes are there. So here is the secondary antibody conjugated. The conjugated with the enzymes are there. So I have already discussed it. We can utilize the 3 enzymes, or it will react with the substrate, or it give the colors. So we can measure it color through the ELISA readers. So in case of indirect ELISA, it is a 2-step ELISA which involve to the 2 binding process of the primary antibodies labeled and secondary antibodies. The primary antibodies is incubated with the antigens and followed to the incubation with the secondary antibody. In addition of the substrate, we will get the colors, or ultimately we can measure it color. On the basis of color we can decide the concentration of the antigens. And third one is the sandwich ELISA. You can see it here is the sandwich ELISA are there. The capture antibodies, here is the antigens are there, primary antibodies and secondary antibodies. You can see here is the sandwich like structures are there of antigens. The antigens, they will bind with the capture antigens. You can coat on the microtiter plate or tubes. Another is the primary antibody, then we can utilize the secondary antibodies. It measures the antigen between the 2 layer of the antibodies, capture and detection antibody. Antigen must contain at least 2 antigenic sites for binding of the antibodies. Monoclonal or polyclonal antibodies can be used. In addition of the substrate, we will get the color, or ultimately we can measure it. The color, we can decide the concentration of the antigens. Here fourth one is the competitive ELISA. In this case, the 2 specific antibodies, one conjugated with the enzymes and other present into the test serum. If the serum is the positive for antibodies are used, the competition occur between the 2 antibodies for the same antigen. Ultimately, we can decide it the how much quantity of the antigen present in the serums. So these are the 4th type of the ELISA are there, that is known as the competitive ELISA. And it is another very important test that is known as the immunofluorescence test. In this test, antibodies are tagged with the fluorescent dye which is detected by to the colored light emissions when excited with a specific wavelength of the light. It is emitted light when the measured bit to the fluorescent microbe equipped with the UV light sources. In this case, we can utilize the dyes. Various dyes are there, like fluorescein dye and phycoerythrin dye, rhodamine fluorescein isothiocyanate FIK, and TRITC, tetramethylrhodamine isothiocyanate. So these are dyes we can utilize in the immunofluorescence test. We can tag your antibodies, or they are able to bind with the antigens. We can see it. The application of immunofluorescence test used for identification of bacteria, virus, and other antigens, and detect immunofluor test to use for diagnosis of the rabies. These are the very important tests are there. So ultimately, rabies virus we can diagnose it. Here is the tagged antibodies or immunofluor test. In this case, the tagged antibodies, which are the fluorescent antibodies, the particular dyes we can utilize in the antigens antibodies. We can utilize for the diagnosis of the rabies in particular animals. The type of the immunofluorescence. Here is the two type of the immunofluorescence are there. One is the direct or primary immunofluorescence. You can understand through this figure. It is a virus are there. They have the particular epitope. Here is the antibodies are there. They have the active fluorophore are there. The primary antibodies labeled with the fluorophores. So we can utilize this type of the system are there, this type of the mechanism to measure the concentration of the antibodies, measure the concentration of antigens. The in this case, the primary antibodies are chemically linked to the fluorophore or fluorochrome. You can see it here. The fluorophore or fluorochrome are attached. The primary antibodies bind to the antigens, and fluorochrome facilitate the visualization of the target antigen under the fluorescent microscope. So for analyzation of this reaction, we can utilize the fluorescent microscope, or ultimately we can measure it. Another is the indirect or secondary immunofluorescence. Here is the primary antibodies are there, another is secondary antibodies, label the fluorophore, we can utilize it, or ultimately we can analyze the particular viruses or the concentration of the antigen, we can measure it. This method use two antibodies. The primary antibody is unlabeled and it is bind to the antigen. The secondary antibodies carries to the fluorochrome and bind to the primary antibodies. So we can utilize it for detection of the particular pathogens and antigens. Here is the fluorescent microscope, which are very important. With the help of fluorescent microscope, we can measure it, the particular pathogens or particular antigens. And it is another technique like immunoelectrophoresis. Immunoelectrophoresis are there. It is a very important technique. We can measure it, the concentration of the antigens. It is developed by the Graven and Williams, and it is blend of the electrophoretic or immunodiffusion. So here is the mixture of the electrophoretic and immunodiffusion techniques are there. The antigen placed into the bell, you can see it here is the bell are there. We can place the antigens of the agarose gel. Is electrophorus for an hour. We can electrophorus of your antigens are there. For our separation into constituent proteins, the antibodies is placed. We can add to the antibodies. Place into the trough. Here is the trough are there. We can put the antibodies, and which is cut onto the agar gel above up to the antigen belts, and diffusion take place for to the 18 to 24 hour, and different precipitin lines are observed for to the different proteins. So we can measure it, the different precipitin line are there. We can find out to the identity, non-identity are there, or partial identity of the particular antigens. We can analyze it with the help of this test. Another is the rocket immunoelectrophoresis. So in this figure you can see it. Here is the cathode are there, here is the anode are there. We can connect with the cathode and anode electrodes. Here is the proteins, here is the bells are there, antigens in the bells and antibodies in the gels. Here is the antibodies, antigen will diffuse it, or we can see the pattern, or we can decide the concentration of the particular antigens are there. The antisera against to the test antigen are incorporated into a growth gel and poured into the slide. The antigen are loaded in a series of the bell into the gel is increase the concentration. Electrophoresis take place in rocket like precipitation. You can see it, it is a rocket like precipitation line are formed, and the highest of the precipitation line increases with the increasing the concentration of the antigen. So if you compare it in this line, you can see it here, the concentration of the antigen is the too high. So we can utilize it for analysis of the concentration of the antigens. And it is another technique, flow cytometry. Flow cytometry is automated technique that used to diverse fluorescent antibodies to stain cell to be separated. So in this case, basically we can measure it the diverse, we can measure it the concentration of the cells are there. The assay can be detect up to the 12 fluorescence and the light scattering molecule routinely. In this assay, cell can be stained with the antibodies. So your cells, we can stain it with the antibody conjugated with the particular fluorochromes like fluorescein, phycoerythrin, or number of the other fluorescent fluorochrome molecules are there. We can utilize it and that make detection easier. For intracellular staining, cell are permeabilized and with the detergent allow to entry to antibody into the cell interior and bind to intracellular component based on to the binding of the fluorochrome facts of a flow cytometer sort and heterogeneous mixture of the cell based light scattering and fluorescent signal from individual cell. Flow cytometer used to laser beams in the series of the detector or the photo multiplier tubes to identify or scattered light or the fluorescent signal of a particular wavelength emitted from signal intact cell following into the single stream. So you can see it, here is the scheme of the facts are there, the laser which are available. Here is the two detection panel are there. They have the positive charge and negative charge. So we can sort out the different type of the cells are there with the help of the fluorescent tag are there. Here is the antibody is tagged. Dear learner, I want to summarize my part second. You have learned the number of the techniques like ELISA, which ELISA we can use the antigen or antibody covalently bound to the enzyme that converts substrate into the colored reaction product. Western blotting is an analytical technique that used to antibody as a probe for the detection and identification and quantification of a specific protein in a complex mixture. And immunofluorescence use monoclonal antibodies attached to the fluorochrome or visualized cell tissues and cellular component. You have also learned a flow cytometry techniques which are we can utilize for detection of the different type of the cell or proteins are there. Dear learner, if you want to learn detail, you can go through the courses immunology. The course code is BZYET 141. You can go through, you can read it, you can clarify each and everything. If you have any query, you can mail me at crm1 at the rate ignou.ac.in. Thank you.
Are you a fresh graduate? Do you want to fulfill your educational aspiration by pursuing higher education in various interdisciplinary and multidisciplinary areas like environmental science, environment and occupational health, philosophy, sustainability science, folklore and culture studies? If yes, the School of Interdisciplinary and Transdisciplinary Studies offers various programs like Master's Program, Graduate Program, Postgraduate Diploma, Certificate and Appreciation Program. The school offers 5 Master's Program. Master of Science, Environmental Science. The eligibility criteria for MSc Environmental Science is Graduate in Science, BTech, BE, Agriculture Graduate, Forestry Graduate and Veterinary Graduate. School also offers Masters in Environment and Occupational Health, Masters in Sustainability Science, Masters in Philosophy and Masters in Folklore and Culture Studies. The eligibility criteria for these 4 Masters program is graduate in any discipline. The school also offers various socially relevant and need-based PG Diploma program like PG Diploma in
Hello and a very warm welcome to all the viewers of Gyan Darshan channel and the learners of PGDPSM program. Today's session is on Introduction to a PGDPSM program, which is Postgraduate Diploma in Pharmaceutical Sales Management. I am Dr. Rachna Agarwal, Associate Professor in the School of Vocational Education and Training, and I will be introducing you to today's session, which is about a particular program. How is it important? What is the utility of this program and why anybody should take admission in this program? So let us begin. What is PGDPSM or why one should do PGDPSM or Postgraduate Diploma in Pharmaceutical Sales Management? PGDPSM is a specially designed program. It is made for those who are aspiring to be medical representatives or any employee of a pharmaceutical company who wants to upgrade his knowledge, skills, can also do this particular program. Now, as we know, different sectors of employment or different professionals require different job roles. They need expertise in different areas, and similarly in this particular program, which is PGDPSM, PG Diploma in Pharmaceutical Sales Management, this program also caters to a particular segment of the society or the professionals, which are medical representatives. And what are the essential qualities or who is a medical representative? We will learn that. But what skills should one possess that they can do this program? That is very important. And in this particular program, we from Ignou are imparting those skills to our learners so that they become successful professionals in the field of medical representatives. Now, medical representative is any person who has to sell the medicines of any pharma company or any big pharmaceutical industry to a healthcare professional. Like, he is responsible for selling of the product, maybe a medicine, maybe an equipment, a new equipment like a BP monitoring machine or kit for measuring your sugar levels, for the diabetics. So, or maybe it can be any implant for that matter. So, any new product in the health science sector which is to be introduced has to be first marketed. Like any doctor or any healthcare professional should be apprised about it. He should be able to understand what this new product is, from which company is this product coming from. So all this convincing power for a new product or a new medicine or an old medicine which is now made for a new purpose or for a new ailment, this is the job of a medical representative to convince any healthcare professional for this particular pharmaceutical product. So, as we see here, who is a medical representative? As I have explained, a medical representative is a person who advertises the medicines of a pharma company to any medical practitioner or a doctor or maybe a chemist also. And the job of a medical representative is very challenging. You see, they represent the company. They are advertising the product of a particular company. So in their particular region or territory, they are the ones who shoulder this responsibility that the positives, the pros and the cons of a product, of a medicine should be well known to any chemist or a healthcare professional. And they are the key point of contact between the pharmaceutical or medical company and the healthcare professionals. They are responsible for promoting awareness about the product, for answering various queries which a doctor or a chemist can have, for providing advice, and also for introducing any new product, as I have explained. So any new product which is introduced first into the market, like if any layman or any common man goes to a chemist, sometimes they just ask, okay, can you give us a medicine for a common cold, or can you give us a medicine for a headache which we are having? So the point of contact here becomes the chemist, and many common persons which we see on in our society, their first point of contact becomes that chemist person. So now if the chemist person or the professional is convinced about this particular product, that yes, you can use this particular product, naturally that product will sell more. He will advertise more for that particular product. So it is the responsibility of a medical representative that he should be able to convince the pharmaceutical shop person or the chemist or the doctor that yes, our product can have these positive points over and above which is already there in the market. Our medicines are having these more positive points. So the entire responsibility of any new product which the pharmaceutical company brings shoulders on the medical representative, and it becomes the job of the medical representative to introduce that product wisely into the market. Now, let us see what we are offering in this particular program, which is PGDPSM. The program details, as you can see on the screen, the program comprises of 32 credits. That is, it is divided into 6 courses, and each course is of a certain credit, maybe 6 or 4, and the total credits are 32. Medium of instruction for this program is English, and the eligibility requirement, as it is a postgraduate program, is graduation degree, and the minimum duration of this program that you can complete in is 1 year, but Ignou is flexible, so it allows maximum 3 years of time to complete this particular program. So it is very flexible. It is a 1-year PG diploma program, but you can also, like if you are not able to give your exams or because of any reason, so you are given a maximum number of 3 years to complete this particular program. The fee of the program is very nominal. It is only 7,400 at the moment, but I would advise you to keep a check about the fees on our Ignou website, that is www.ignou.ac.in. Now, the main objective, as I have explained, the responsibilities of the medical representative, the main objective of this particular program is to impart knowledge and skills for selling of a product. That is, so here in this particular program we are imparting knowledge about pharmacy, and also skills and knowledge about the managerial aspects, that is sales management and marketing. And what are the requirements for completing this particular program? I will explain here. Since there are 6 courses in this program, so the requirement of the program is to submit 6 assignments, that is 1 each for each course, and you have to appear in 6 term and theory exams. There is no practical component and there is no project in this program. So all what is required is submission of assignments and appearing in the theory exams. Now, when you have to write the assignments, when you are writing and submitting the assignments, there are some conditions which you should keep in mind. That is that your answers have to be written by hand. They should be handwritten. Like nowadays, learners are referring to various sources on the internet. So they would use ChatGPT or Google, and they can get more knowledge. But that knowledge you cannot just copy paste. You have to write it by your own, in your own handwriting. And they should not copied also from your text material as it is. This is also, we do not want. You should learn from the text material and you should write in your own language. As I said, answers in form of photocopies or scanned in any form from any source are also not acceptable, and the assignments should be submitted at your study center or the regional center before the last date which is mentioned in each assignment. So by way of assignments, the purpose is that we are imparting you knowledge. We are keeping a check that you are going through these study materials. Also here we are giving soft copies of the study material which is available on Empc Ignou website, and you can also download your study material soft copies from there. Then you may also see the videos which are related to the various topics which we keep producing from time to time. Many such videos are available on the YouTube, and the name of the YouTube channel is Empc These videos are also provided by the program coordinator, that is myself. I will be providing you once you have registered in this program, and you can learn about any topic from these particular videos which are pre-made. As far as the program structure for this particular program goes, I would like you to see this table. That is, the entire course or the entire program is divided into 6 courses. Here you can see the first course is on Introduction to Anatomy, Physiology and Pharmaceutical Chemistry. Second, on Pharmacology and Toxicology. Third is on Pharmaceutics. Fourth is on Drug Regulatory Affairs. And the last 2 courses are on Management. That is, Introduction to Basic Management Principles and Sales Management or Marketing Management. So in all, you can see on the right side there are credits mentioned against each. So one course, the first course, as you can see here, is the 60 credits. This denotes the weightage of the particular course. So here we usually consider that 1 credit would involve 30 learning hours. So you can multiply and accordingly plan your study hours. Now, if I have to explain in brief, you can see the first course, which is on Introduction to Anatomy, Physiology and Pharmaceutical Chemistry. It comprises of various things, various text materials which are on human body systems. Like the human body systems may be circulatory system, skeletal system, respiratory system. So each and every system here is explained in detail. That is, what are the various in circulatory system, blood vessels, or what is the cardiac cycle, what is the heart, how does the heart function, And in skeletal system, we have about the joints and various bones, and our skeletal system, what are the, how they are formed in the body. See, this entire knowledge of the systems is very essential in the human body, because ultimately we are dealing with medicines, and these medicines will act on our body. So the first preliminary course should be on the basics of our body anatomy, and some introduction towards the physiology, how our systems work, what are the various processes, metabolism of the food, and what are the various reactions that are going on in the body, so that you know the basic fundamentals of the human body. Then only you will be able to understand that when we are giving some medicine in the body, or when we are injecting an injection into the body, or if we are putting an implant, how that medicine will be taken up or act in the body. So a basic knowledge about all these systems is given in the very first course, and you can see all digestive system, endocrine system, about the glands, excretory, reproductive system is all very well explained. It also gives you a brief idea about the diseases and disorders which are related to each and every system. And apart from that, the diseases and disorders with regard to the environment, like maybe it is a communicable disease, which may be airborne, waterborne, like typhoid, cholera, how it happens, what are the effects produced in the body, and also how one can counter those effects, which medicines are working on that. So vector-borne diseases, airborne diseases, waterborne diseases, and certain non-communicable diseases, that is cardiovascular disorders, hypertension, angina, digestive system diseases, respiratory system diseases, pneumonia, bronchitis, asthma, they are all explained very nicely in the very first course of this program. Then we have certain nervous system diseases which are now becoming very prevalent, like Parkinson's, epilepsy, Alzheimer's, and also our body's immunity. What is immunity? Whether we are, certain individuals or ethnic groups, as we say in the scientific language. Ethnic groups, you understand that a person coming from Africa belongs to an African ethnicity, and we from Asia belong to a different ethnicity. So certain group of people are already having certain immunities in the body. So these are natural immunities, whereas some new diseases which are coming up, they require antibodies to be produced in the body. So these immunities have to be acquired from outside sources, maybe through injections, maybe through medicines. So how our body can be equipped and become more stronger with these immunities. So this is also explained in this first course. And certain pharmaceutical chemistry topics like isomers, buffers, about the basic knowledge, because we are dealing with medicines, are also explained here. Sterilization techniques and some other blood related and disorders and cancerous diseases. So this is about the first course. Now all this knowledge is provided, which is not only beneficial for any medical representative, I would rather say it is good for any layman. Everybody should have knowledge about their own system, and this is relevant for each and every person, I would say. Second course is a little more towards the pharmacy, and it deals with pharmacology and toxicology. So here in this course, we are explaining the general action of the drugs in the body. Like if we consume any drug in the body, how that drug goes to the target point, whether maybe it is targeted for a nerve disorder or a cardiac disorder. So how it acts in the body, what the routes it follows, and what is chemotherapy, how the chemotherapy drugs act in the body, what is poisoning, what are the various drug interactions which are having or taking place in the body. So this all is explained in the second course, which is on pharmacology and toxicology. Now, in the third course, which is on pharmaceutics, we have given an idea about the various kinds of drugs which are available. Like you have, sometimes you see the drug is in the form of a capsule or a tablet, or it may be in the form of a syrup or elixir or drops, because different parts of your body require drugs in a different manner. Like for eye flu, nowadays we are seeing that a lot of eye flu cases are coming, so we require ophthalmic productions which are sterile and isotonic in nature, and these drops have to be put in the eyes directly. So what are these various kinds of dosage forms? This particular course gives you an idea about all these different types of dosage forms and how we can calculate the dose of a drug. See, for any infant or for any child, the dose of the drug varies. Now, what is the criteria of that particular dose? Why it varies is because not only that the child or an infant is of a smaller age, but the main criteria why it differs from an individual to individual or from a child to an adult is because of the body weight. So according to the body weight, how we can calculate the dose of any drug? This particular calculation and this particular exercise is explained in the third course, which is on pharmaceutics. And what is the correct way of delivering a drug? Now, we will also educate you about the different routes that a drug follows and what are the different routes of giving the drug into the body. But not only that, what is the correct way of drug delivery and how we can maximize the benefits of the consumed drug. So, all this knowledge is given in the third course, which is on pharmaceutics. Then action of drugs in the treatment of diseases, nutritional deficiencies, and how to take the right kind of nutrients to be healthy, and what are the supplements that a body requires to promote health and well-being. This is all explained in the third course, which is on pharmaceutics. Now, the fourth course, which is on drug regulatory affairs, it explains various acts, rules, and procedures for launching of any drug into the market. How the pharmaceutical industry has evolved. What are the different phases of evolution of the pharmaceutical industry, and how a drug comes into the market. Like you must have heard of a term called clinical trials. So these drugs are first subjected to experiments in laboratories with human volunteers. So what are the different phases which a drug has to go through before it comes into the market? This is all explained in this fourth course, which is on drug regulatory affairs. Then pharmaceutical industry, about the pharmaceutical industry itself and what are the various new drugs. How a new drug is launched into the market. What are the procedures which have to be adopted? What are the stringent laws or the norms which we have to follow for bringing any product into the market? This is all explained in the fourth course, and how the various pharmaceutical laws and acts and legislation has come up. Like for example, in this particular course, we are giving you an insight into the some of the acts and the rules, like Drug Cosmetics and Acts and Rules, MTP Act. So various acts and rules which are relevant and which come under the governance of the drug cosmetics and acts and rules. They are also explained in this particular course. Now the last two courses, as I have mentioned, are on management, because since this is a blend of both pharmacy and management, so we are giving you an insight not only into the pharmaceutical part but also about the managerial aspects of managing a product or a marketing of a product. So in the fifth course, which is on Introduction to Management, we are giving you insight into the basics of management, like what are the tasks of a professional manager, what are the managerial skills that one should possess, and also what are the responsibilities of a professional manager. Then the various aspects of management like planning process, decision making models, techniques and processes, organizational culture and managerial ethos. Then certain things which being from a field, since you will be marketing the product in the field, then certain things which are beneficial to you or as an employee or to your employees would be stress management. Then how to train your staff or an employee, training and development, then manpower planning, controlling, leadership styles, and other group dynamics. They are all explained in the 5th course, which is on Introduction to Management. Then lastly, the 6th course is on sales management, because that is the ultimate aim of any medical representative. So what are the selling skills that a medical representative should possess, which includes communication skills, presentation skills, negotiation skills, and sales display. These are all given in the 6th course. Then mostly we see that the task of a medical representative is to sell, which involves a lot of field work. So the skills which are required to do so are discussed in this course, which is on sales management. And introduction to various marketing and its concepts, personal selling, sales processes, and introduction to sales management. Then certain communication skills, negotiation skills, and handling and promotion of tools is also explained in this particular course. Lastly, I have one very frequently asked question about this program, which is whether one can open a medical shop or a chemist shop after doing this program. So, viewers, the answer is no, because for opening any medical or a chemist shop, we require license, and that license has to be obtained from Pharmacy Council of India. And the minimum qualification for obtaining any license for opening a medical shop is diploma in pharmacy. So this is a PG diploma in pharmaceutical sales management. My request is to all our viewers not to confuse this program with D-Pharma, because this is for tailor-made for pharmaceutical employees, company employees, and for medical representatives. So for any further queries, you can visit our website, Ignou website, and the program is housed in the School of Vocational Education and Training, and you can also see details of this program once you visit the website, either the common prospectus or our school's website, which is SOVET. And admissions are going on, and the date is extended till 31st of July. So I would encourage all my viewers who are interested and whose friends or colleagues are interested, they can join this program. Thank you very much.
We wear clothes on a daily basis. But have we ever wondered, where do our clothes come from? Who makes our clothing? And who are the people and the process responsible for the whole manufacturing of our clothing? If you want to know more about this, Indira Gandhi National Open University from the School of Continuing Education has launched a program which is Certificate in Apparel Merchandising. There are many benefits after doing this program. You can become a designer in various fields. It can be fashion designer, apparel designer, costume designer, or even a textile designer. One can also work as a fashion consultant. Research and development is another field which one can take up after doing this certificate course. Going into academics is another perk after doing this program. Getting into fashion houses or fashion studios as an intern is also a very good option post doing the certificate in apparel merchandising. The basic eligibility of the program is 10 plus 2. The duration of the program is 6 months to 2 years. The program is also available for both the admission cycles, which is July and January. The exams are conducted twice a year, in June and December. So what are you waiting for? Join this program and get an enriching knowledge in the field of apparel merchandising. And to do so, you can log on to the official website of the university, www.ignou.ac.in.
Welcome learners. I am Dr. Pawan Kumar, Assistant Professor, School of Sciences, Ignou. So today we shall, I am here to present a basic introduction to the course Computer Graphics. This course is offered in our MSc Mathematics with Applications in Computer Science program, and this course is offered at the third semester. It is worth 2 credits. So in this session we are going to discuss what exactly is this course all about. The title of the course is Computer Graphics. So we shall see here what does the computer graphics mean, what are its applications in real life, and what are the course contents you will see here, and also we shall see whether do you need any other knowledge before learning this course. So let's start with the introduction. So the first thing is, often students are worried about, or they ask questions like, what does actually the computer graphics means? So when you look at any computer generated image or picture or any computer generated video, you see that some computational tools, techniques are involved, and those tools actually produce such images or videos. So computer graphics is all about producing, managing, or playing pictures, videos, or just produce interactive graphics. So let's look at some pictures. Look at this picture. You can see that there are two dinosaurs as if they were trying to fight each other, and this picture is not actually real, as you must know, but it looks like that it has been some scene is there and it has been captured like that. Okay, so this picture has been generated by computer using some computer tools, some techniques. Likewise, look at this picture, some ape. We shall show you some videos where you can see that how these videos are created using computer graphics. So let's look at this car simulation video. So here you can see that some car is moving. Here you can see that, okay, so once such videos are created using computer graphics, the creator knows certain things how to. So there are basic things here. You can see that there is some car models are there. There is some scene that has to be built, and then once these objects are created, they have to be put into motion. Let's look at some more videos. Here you can see that the motion is this shows that how some animals attack on their prey. So here a snake is just attacking on its prey, which is a rat. Here you see that this video has been created in such a way that it can show the motion in slow. So here, okay, so this is just one of the videos which has been created by computer graphics tools. Now look at this video. Here you can see that some dinosaur is following a man, and now what happens here, when you create such videos, you need to imagine what you have to create, and once you have looked at what are the things that you need beforehand, and then you build the object, and once the object is ready, you apply different transformations on the object to create motions or animations. So computer graphics deals with all kind of such picture generations or video generations, and we shall see what are some applications of computer graphics in other fields. So, computer aided design is one of the field where computer graphics is used heavily. Suppose you want to design the interior of some building, you would like to consider some factors like how the building would look like from inside, what should be the texture or paint on the walls, where the windows, what kind of uh, scene you would like to have on certain windows, or what kind of patterns you might put. So all these things can be beautifully created using computer graphics tools with the tools that are available in market. So computer aided designs, CAD designs are some softwares which can help you create certain computer assisted images. Suppose you are creating some car model. How would you draw it? The basic first hand information that you will collect about the model is that you would see that how does the model look like from the outside. So suppose you have to create some car model, so you will look like, okay, you will just draw certain lines, and by hand when you draw, you just look at, okay, how to draw the front part, the back part, or how does the model look from the different angles. So you would just draw a rough diagram. Here in computer graphics, this terminology is called wireframe model. So you just create some wireframe model, and then once that model is ready, you would like to put certain like shades to it, or you may like to create the views. How does the model look like from its interior, from inside? So for instance, you would, once the outer model is ready, you would like to see the inner model, what is its capacity, say how many seats do you want to put inside the body, how much space you want to put for the front part, for engine, or for back part, how does the interior look like. So all these considerations you will take into account. So, and then you would say possibly look at some other individual parts. For instance, you would like to create or view how does the engine of the model look like. So this just picture shows some cross-sectional view of a car engine. So when you have to show these kind of pictures, it is the computer graphics tools are very handy. And okay, another application that we often see is presentation graphics. So while suppose you are you have to present certain things to your audience, the best way is to use certain pictures, right, instead of using text material. So suppose for instance you are some smart city project manager, you would like to present your project to the audience. So what are the things that you would like to put into your slides? Maybe you would see that, okay, these are the things that a smart city would contain. Maybe how does the society is located, some infrastructure, energy resources, and where the healthcare should be, whether all the other ingredients are there or not. So you can just produce any kind of presentations using computer graphics tools. A lot of tools are available in market. This is just like some picture created using computer graphics. This is just some electric vehicle presentation. Maybe say while you are presenting some electric vehicle promoting, maybe you would like to see what are the key factors or key benefits you would like to present. Another important area where computer graphics is heavily used is computer art. So earlier artists were used to create paintings or pictures using hand, but nowadays a lot of computer graphics tools are available in market that can assist you in creating or designing beautiful pictures or paintings or artistic imaginative ways to create different kind of paintings. This is just some computer art generated picture of some mathematical shapes. Like this is also some mathematical shapes. So apart from computer art, the computer graphics is heavily used in movies and entertainment industry. So nowadays you can see that movies are heavily dependent on computer graphics tools. In imagination or imaginative ways, you can see that how does action scenes are performed which are not real and which cannot perform in real life. So but using computer graphics they can be shown on the screen, like many Hollywood movies. In fact, nowadays almost every movie uses certain kind of computer graphics tools. These are certain some pictures taken from certain famous movies. All these have used computer graphics. So this is one of the Bollywood picture creature which you must have seen. So it is, this creature has been actually created using a lot of computer graphics tools. So after knowing now what are the applications of computer graphics, you would be interested to know what this course offers to you, what the things you would learn in this course. So I would like to now briefly explain you the contents of this course. So as I have said, this is a 2-credit course, and the course only gives you certain basic graphics tools, which and you will see how to create certain basic graphics pictures and some kind of animations, and you will appreciate how this course is built into using the programming languages that we have kept in our program, like C programming language and other tools like Scilab and other MATLABs, etc. So the contents of this course are there are 3 blocks in this course. Block 1 just discusses the fundamentals of computer graphics, and block 2 discusses 2D transformations and clipping, and block 3 tells you about 3D transformations and viewing. So let's look at each block contents separately. Block 1 is divided into 3 units. Unit 1 is just about basics of input-output devices. Unit 2 just tells you what are the generating graphics primitives, how will you generate 2D images like point, circles, or some conic sections or straight lines. And then in Unit 3 you will see some filling algorithms. Filling algorithms just used to fill areas or regions like polygons. You suppose you would like to fill some polygon with the color. So you would see here a number of algorithms. You would need to know what are the techniques that are involved in area filling or region filling. So now in block 2 there are 2 units, and which is 2D transformations and clipping algorithms. So 2D transformations are actually the transformations in 2 dimensions, like what does say translation, rotation, or scaling, or shear other transformations. So these transformations are actually you must be familiar with, while or certain basic terminology are used here, and to just illustrate how these transformations are used in creating or transforming different models or objects. The Unit 5, which tells you about clipping algorithms, is actually gives you only a couple of algorithms where you will see once suppose you have created some scene using some algorithm, right? You would like to show only certain part of the scene, so other portion of the scene must be clipped off. So what are the procedure for clipping? You will see in this unit. And then the last block, which is the third block, it just gives you a glimpse into what are the three-dimensional transformations and viewing transformations involved in computer graphics. So unit 6 is just gives you just list you the 3D transformations like translation, rotation, scaling, etc., which we have also studied in unit in the first block. These are actually the transformations which are just generalized from 2 dimensions to 3 dimensions, and these are very important because once you have created an object, you need to know that need to know how to transform one object from one place to another, or you would like to create multiple copies of the same object to create some scene. So these transformations are quite useful. And the last unit is about display methods. It just tells you what are the different display techniques are used in computer graphics, and you will just only see some basic information about them, and some algorithms are there. So this is all about the contents of the course computer graphics, which is offered in this MSc program. So what are the things that you need to know before learning this course? So some of the basic knowledge about analytical geometry is just needed, because about 2D and 3D transformations you would need a lot of use of these things. So you need to know what are the basic concepts involved in analytical geometry. Apart from then, you also need to know some concepts from vector calculus, and of course finally you need to know some programming language. So in this course we have used C programming language, and which you all must have studied in your first semester course, programming and data structures. So this program, apart from these three things, some understanding of basic calculus is good, like what are the curves, what are the tangents, and these things are slightly needed, but that is all about this course. So apart now, once you see when enroll for this course, the course material has been presented in the 3 blocks which I have described here. So apart from studying this study material, you would like you have to undergo some practical sessions at your study center, and these sessions are just contain certain practicals which have been based on the concepts or algorithms which have been described in the material. So you have to go through all the exercises that in the text, and then you can work out the practicals at your study center, because to clear this course you need to have there are 2 components, theory as well as practicals. So in you have to clear both the components to pass in this course. So apart from the study material, you need to know some basics about the OpenGL. So what exactly this OpenGL? It is actually just a software interface to the graphics hardware. You know that every PC that can generate compute images has involved or has just equipped with some software, and to produce the graphics output or graphics images, the software that we use is OpenGL. It is actually the software toolkit that we use. Apart from this, the programming language editor that we use is code blocks. So there are, in the code blocks manual, you will find that how to open a OpenGL project, and you can write computer graphics programs using that interface. So this OpenGL is available in all the major platforms, Windows, Linux, and Mac operating system. So now there are certain, apart from the study material, you can also look at some standard books for computer graphics, and these books are Computer Graphics C version. It is by Hahn and Baker. Another one is Computer Graphics using OpenGL. It is by Hill and Stephen Kelly. And another book which is also quite interesting is Computer Graphics Principles and Practice in C. So it actually uses C programming language. It is by Dam, Fiener, John. And apart from these three books, there is also OpenGL programming guide, where it just lists all the basic or commands for creating computer graphics programs. So these things are there. So once you register for this course, actually you need to have good understanding about C programming language, because writing computer graphics programs involves a lot of data structures. For instance, you must be very much familiar with well versed in the stacks and queues, how to create linked lists and how to use them in your program. For many algorithms in computer graphics are efficient just because of they use high level data structures. So a good programming skills are needed in this course. So you must have already passed in your C programming and data structures course. You can revisit that course if you find it difficult. So this is just all about the introduction to this course. The course material you can just go through, and you will find that this course has a lot of interesting references also there. You can go through online tutorials where you will find some questions and some practice sets. So thank you.